Hi, my name is Adam Steinbrenner. I'm an assistant professor at the University of Washington. Thanks to the organizers for the opportunity to speak today. And I'll be telling you about my lab's work on using plant genomes to home in on plant immune functions, specifically how a single immune receptor gene has evolved across 50 million years of legume evolution. And we know that plant immune receptors are among the most rapidly evolving parts of the plant genome, but I'll be telling you a little bit of a twist on that story today, whereby we talk about a process by which a plant immune receptor acquires fixation in both its genomic stability and its recognition functions over evolutionary time. A little bit of plant immunity to start the talk. I'll be talking about pattern recognition receptors in the leucine-rich repeat family, the largest family of PRRs in plants. These have a leucine-rich repeat ectodomain and signal through intracellular kinase cascades to carry out plant immune functions and activate inducible defense responses in plants. One well-known PRR is FLS2, which detects FLAG22, an epitope from bacterial flagellin, but there are many other PRRs in plants. Tomato CF2 protects against leaf mold, RLP42 in detection of botrytis associated proteins. And then we recently described a PRR called Inceptin receptor that detects peptides in the oral secretions of chewing herbivores. So this concept of detection of a non-self pattern applies to defense against bacterial, fungal, and herbivore pests and pathogens. These PRRs come in two flavors. There are receptor kinases, which have an intracellular kinase domain, and receptor-like proteins, which lack one. Receptor kinases are more numerous. There's many hundred per genome, and they serve both developmental and immune-related roles, whereas RLPs are fewer in number, but really seem to be the rapidly evolving end of the cell surface machineries that detects these extracellular pathogen patterns. So, for example, in our recent review, we characterized the RLP repertoires from both Arabidopsis and tomato. And you can see that these two genomes have completely unrelated clades of these receptors where there's been huge species-specific expansions of RLPs. And these include some of the important functionally characterized RLPs that I've told you about before. So RLP42 is absent in tomato and CF2 is absent in Arabidopsis. So these make them attractive traits for transferring across different plant families. And the processes by which they evolve are important to understand. We have a family-specific PRR in my lab that we study, and it's called Inceptin receptor. It detects an 11 amino acid fragment, peptide fragment in the oral secretions of chewing herbivores that's derived from degradation of a host ATP synthase, which gets regurgitated back onto the plant and detected by the plant immune system. We knew that Inceptin peptides were only active on certain legume species. So this felt like a situation where there might be an evolutionary acquisition of a new peptide sensing function specific to legumes. When we identified INR, we of course looked across different plant genomes and noticed that we discovered it in cowpea, but there was a homologue at the same locus in common bean, in mung bean, but not in cold season legumes like medicago. So this suggested that this was specific to certain legumes, but we still didn't have enough phylogenetic resolution to tell where this gene insertion event occurred and the process by which it acquired peptide binding and signaling functions. So how is a new PRR born was the question going into this study. A postdoc in the lab, Simon Snook, took on this question. And his first task was to screen legume diversity for Inceptin responses. We collaborated with Ashley Egan at Utah Valley University to come up, legume systematis, to come up with a great species tree of legumes, which includes not just cultivated species, but semi-cultivated and wild species such as guar bean and soybean. And then what Simon did is grew up these species and tested for their ability to respond to Inceptin peptide. We're using a very simple generic defense marker, the production of inducible ethylene. And I'm showing you here on this axis, this is the ratio of plants treated with Inceptin peptide versus just water as a mock. So many species, if this ratio is 1, there's no Inceptin-induced response. And there's many species in this category, this red category. But then we also successfully measured an Inceptin-induced ethylene response in many different responsive legume species, including in cowpea, where the receptor was first characterized. If you map this responsiveness back onto the species tree in blue here, we can see that there's a very clear pattern whereby only phasialoid legumes respond to Inceptin. So there's a single hypothesized origin of Inceptin recognition at the base of the phasialoid legumes. And then we also see there are secondary losses of recognition in various clades here, including soybean, of course, a major crop. And so we were further interested in understanding this pattern and whether diversity at the Inceptin receptor locus correlated with this pattern of responsiveness across the legume family. For this, we needed additional de novo assemblies. We collaborated with Todd Michael at the Salk Institute, and his postdoc Brad Abramson, to put together new long read-based assemblies at key nodes in the species tree. And this was a really successful and fun process of generating some high quality new genome assemblies to investigate one specific locus of interest. This really, they helped, Todd and Brad really helped facilitate this, but it did not take too much data, 20 gigabytes of nanopore data, and then Illumina data for polishing the assembly to generate really a pretty good working assembly from which we could pull RLP homologs from these different species. So for example, from Hylodesmum podocarpum, we were able to extract a single continuous INR locus with 2 flanking well-conserved transcription factors, and then a variety of RLP encoding genes between, kind of interspersed between various repetitive elements of this complex locus. If we put all of these loci, INR loci in sort of a Synteny view across the NP-AAA papillinoid legumes, we start to see some interesting patterns about PRR emergence in evolution. So here we're looking at outgroup legumes, in the cold season legumes, including Medicago, which I mentioned, but also a new assembly here, Centrosema tetragonaloba. And we can conclude that some of the, all of these cold divergent legumes lack any INR paralogs. So this seems to precede the origin of any receptor genes at the locus. If we move into the militoid legumes here, this includes jack bean and rosary pea, we see that we start to see RLP homologs at the locus. So this really strongly suggests that there was a single ancestral gene insertion event in the militoids that seeded the existing RLP diversity at the locus today. And then once we move into the phasialoid legumes, which includes Inceptin responsive species in blue, we start to see a large degree of copy number variation. So anywhere from 0 to 7 copies, depending on the species. And notably, there's stability with only 2 copies within the Phasiolinae, a 13 million year old clade of legumes, which includes common bean, cowpea, and mung bean, where there does not seem to be any significant copy number variation. The locus seems to have stabilized within the Phasiolinae. What we can do with all these RLP genes is extract them all out and make a gene tree phylogeny to look for functional patterns. And specifically, we're comparing all these homologs to the functionally characterized cowpea homolog up here that we first described in 2020. What we can see is that there's a single clade of receptors that seems to cluster around this functional copy circled in blue here. And we call this the INR clade. And specifically, this clade includes a single gene from every Inceptin responsive species that we studied. So we think that this strictly correlates with the ability of any given legume to respond to Inceptin, is the presence of an INR homolog from this clade. The way to test this is we use a heterologous test system. We can express the receptors in tobacco, in Nicotiana benthamiana, and then measure an Inceptin-induced Ross burst. So here we're looking at the production of reactive oxygen species 10 minutes after peptide application to tobacco plants that are expressing the Inceptin receptor from common bean in this case. And so if we express INR from the INR clade in tobacco, various INR homologs are able to confer an Inceptin-induced response consistent with the single clade of functional INR homologs, whereas diverse paralogs from common bean, from cowpea, and from soybean are not able to confer this Inceptin-induced response. Using this sort of this broad functional validation of various homologs, we were able to also ancestrally reconstruct Inceptin receptor function. So the node here at the base of the INR clade, we reconstructed that ancestral homolog, and it's able to confer a ROS burst, whereas if you go one node further, the INR-like ancestor of this entire set of RLPs is not able to confer this. So this really gives us a lot of resolution into functions, into specific changes that mediate gain of function in peptide binding and signaling. And so ancestral reconstruction of these receptors really does narrow it down to just 16 amino acid polymorphisms in the ectodomain of these proteins that gives Inceptin receptor a novel functionality. We also see an interesting pattern whereby these species that have secondarily lost INR, their receptor copies are more similar to one another than they are to any other species in our analysis, which suggests that their loss of INR was mediated by either a deletion and then rediversification of the locus or gene conversion. Both cases are consistent, and with this case where it has lost the functional INR clade member, the blue copy here, and is instead seated only with INR-like homologs. And this is true not just for glycine, but for hylodesmum as well, all the orange copies down here. And so here's our model at the end of this kind of functional genomics study of how a single PRR acquires a new function over a long period of evolutionary time. In non-militoid species, we don't have any RLPs at the INR locus, but about 32 million years ago, we get an insertion event that leads to unstable copy number variation at the locus. But upon acquisition of an INR function, that really stabilizes the locus either with or without paralogs, but species tend to encode that single INR homolog at this stage. And then we do have cases where single species have lost a functional copy, for example, in soybean and hylodesmum. So there are cases of independent secondary loss of INR, potentially due to polyploidy or genomic instability. And we think this is the case for one receptor, but remember that there are dozens to hundreds of these per genome. And so we think this will inspire a phylogenetic sort of search image for useful PRR gene traits. And I'm calling this sort of not too hot, not too cold, where for a given species complex, we're looking for receptor genes among their repertoire of dozens to hundreds, genes that are not too polymorphic within most species, so they're very stable within species that do encode them, but they're also not too cold. They're still evolutionarily novel relative to outgroup species, species E and F in this case. And then we're also looking for this feature where these useful receptors happen to be absent in a target crop, soybean, for example. And so one project in the lab is to move INR back into soybean to see if this confers useful caterpillar pest resistance. And so I hope I've convinced you that technically a family-wide genomic survey is really straightforward using cutting-edge long-read genomic technologies and yields functional insights for loci of interest. And I would also highlight that this strategy of family-wide characterization of immune receptors really contrasts with the current way we look for our resistance genes, which is to look for basically highly variable genes that confer resistance phenotypes, but that may be less evolutionarily stable in the long run. I'd like to thank my lab, especially Simon Snook, who spearheaded this work, our collaborators at the Salk and Utah Valley University, and our lab is funded by the Royalty Research Fund at UW and the NSF. Thanks, and I'm really looking forward to your questions.